Right. So this is the point in time when there's this very bright light shining on me right now, and we're gonna turn it around and focus on the panel. So, as I mentioned, we solicited questions in advance, and I have gone through these questions and prepared a list here. And we've done this in the past, and the whole intention here is to I'm sure many of you have the same questions, so we wanted to get as many of those possible addressed, and also have a chance for the community the wider community out there to ask their questions. Actually, and while all three of you were talking, I came up with one question that I'm guessing some of you are asking: what exactly does IND stand for? So if I can answer that question, I'm guessing some of you may have been wondering that. An IND is a investigational new drug application. And it's a very involved document it's all filed electronically, but it's a very large document that is basically where you put all your information together and you present it to the FDA and present it to them for review. And it is a critical step that is necessary before a compound can be evaluated in human studies. And so that in all three of these cases, that's going to be a critical step, and we're pre-IND filing in all three of these cases today. So let me start with some of the questions. The first set of questions are shown here. And I've tried to group these into sort of cohesive units that flow together. And if the panel, some of you want to take some of these, but the first question was what genetic subclasses will be targeted? And Matt, I think you mentioned that in your case, we're pretty much targeting all the different genetic subclasses. Eugene and Jody, what are your thoughts on the different genetic subclasses? And
yeah, we'll be targeting the deletions to start with.
Yeah, we're most likely going to target all of the.
Are you on?
Yes, on.
No, they're both on. It was picking it up.
We are planning to study all of the genotypes of AS.
Okay. And what ages will be targeted? So, Matt, you've talked about yours. We want to.
Just one caveat is that you will need to have an identified. As we know, there's a certain population that do not have a identified genetic cause that actually have Angelman syndrome, have the pathognomonic sort of EEG signature. But if you don't have the genetic mutation, well, unfortunately, at least at this stage, that would not be eligible. And I think that would probably be consistent with my colleagues as well. Now, as I said before, our initial trial will involve 16 and above, and then we will go back into a much younger population. How young that will be, ideally would like to go to the point of diagnosis. The biggest challenge for us, of course, if we're enrolling infants of 6 months or 9 months, is really what our endpoints will be. But there's a precedent that you don't need necessarily to have the same endpoint for each cohort within your trial. In other words, you can have one endpoint for children who have a certain function or certain age, and you can have another endpoint for an older population. So we're hoping, ultimately our goal, of course, is to have the broadest group and every individual angel being eligible for the drug. So that requires you to, if you want to get that, what we call the broad label, so when the FDA approves a drug, it doesn't approve the drug for everything, it actually defines the subset of patients that you can market and sell your drug for. We want to get the broadest label. That means getting the broadest population in our clinical trials. But we're starting conservatively, and then we'll move back into the pediatric group.
Yeah, I would echo that statement. You're looking for a broad coverage, but your first study is a safety study, so we would be in a phase 1. So usually, it's going to be an older population to start with, and then move it down into a younger population. Although, as we've heard from the scientists and as everybody hears, you want to go as early as possible, but you certainly have to get through the initial phases with going through the early evaluations and then bring it down in age. So we would be very much targeting what Matt was saying. The other issue is, again, on the endpoints is, what is the appropriate endpoint, and that too might change as you go younger and younger in age. And typical drug development, drug discovery usually does move its way down.
Yeah, I completely agree with my colleagues. I would only add that oftentimes the actual age range is kind of negotiated. You really don't know. You can propose something that makes sense, and you could be told by FDA that it doesn't make that much sense to them, and you need to move your strategy or change your strategy slightly. But the general approach of starting your safety study with the older population, typically adult, but not always in rare diseases. And we've certainly seen this with SMA, where, again, our first study was in older patients, but very rapidly, once we saw good safety and tolerability, good feasibility of the injection in that population, we progressed into study in infants. So it oftentimes is a kind of a give and take conversation with the FDA. What they really want is thoughtful approach, careful approach that is thinking about patients first, but it also has to be, they always think in terms of what is the risk and what is the potential benefit on the other end. So that's usually a fine balance. If you are studying a population in their mind that is not likely to benefit, that may not necessarily be acceptable to them. They may actually change direction and say, well, we think you should go younger, or vice versa. They can say, we don't think that based on what you've collected in animals that the safety package is strong enough to go into younger children. We want you to really start at the other age, at the other end of the spectrum. So it oftentimes is something you propose, but you really negotiate and discuss with the regulatory authorities.
So we've talked there about the ages that will be targeted in the trials, and the next question is related to this, and maybe Eugene, you can start off on this one. Are the treatments anticipated to work only in children or also be beneficial to adults? And maybe if you could comment on some of the current vision of that.
Yeah, that's
that
a,
issue.
That's a tough one, and it is always a tough one in neurodevelopmental disorders, and we think that there are some aspects of the disease that are going to be more responsive in younger population who haven't really had the haven't had to live with the pathology for years, if not decades. But it's quite possible that UBE3A restitution or correction could address some of the significant impairments in adults. We just don't know, and this is why we need to study more than a single animal model. What we've done in SMA, again, was very instructive because we started with a very severe animal model looking at just survival. And once we established that, we moved into sort of a milder phenotype that had more relevance maybe to older human population with SMA, where we could show also some improvements. And that gave us confidence that we could actually address more than just a subset of patients. What's going to happen in Angelman? I don't know yet. We're still in that learning phase of seeing what real effect antisense has in this disease.
And Jody and Matt, your thoughts. So your treatment approaches, are they anticipated to be more effective in young or old, or where are we? What is your view on that question?
Yeah, I would echo what Eugene just said. We don't really know. If you I'll stop it. Okay. We don't really know. We would, you could think that in fact it could be beneficial. It might be more beneficial younger than older, but it doesn't mean that it's not going to benefit at older levels. And so we'll definitely have to do more work on that.
Matt, your thoughts?
Yeah, I think the general principle is that, it's relatively easy to intervene and make a change in a very, very young child. For example, in pediatric epilepsy, the patients with very, very severe seizures, you can do a hemispherectomy, chop out a quarter, even half the brain, there's recovery. You would never be able to do that in adults. So the degree of recovery of function is always going to be greater in a younger child. And certain strategies are more targeted to that. And the same caveat, there are certain drugs that actually work greater on those with the greatest disability, and you see the greatest benefit. We do not know with our compound whether we will have greater activity in those least effective or those most effective. my intrinsic bias, it's more likely to have greater long-term gain. The moment you start any degree of recovery, you also open up the ability of that child to engage with the environment, and the earlier you do that, the trajectory of the disease will change if you make an improvement. So even if your drug is equally effective as young and old, you're always going to be biased to having a greater impact with a younger child, because you'll change the trajectory, the natural history of how the disease will advance. So I think the general principle is the earlier we can go in, the better as a general principle.
So the next question is, are there any benefits specific to the UPD group? And then I add also any other group. So we have various different genetic subclasses. Are they different genetic subclasses anticipated to respond differently? And Eugene, maybe, I know this is an issue or a specific question concerning the ASO approach. So
Yeah, I think in theory that certainly makes sense, and this is what we believe. But again, proof is in the pudding until you actually look at the data. We've been surprised a lot of times before where it makes very little difference that what type of mutation, or whether it's missense, nonsense, and we don't really believe that if the impact is large enough that you will see meaningful differences amongst different genotypes. So you should see correction and all the benefits of that correction on a population level. But will there be small differences, and will some patients respond better? That's always the case in clinical trials, and oftentimes, more often than not, we can't explain it. Some people respond better. In some instances, it relates to the levels of the drug. In most instances, it doesn't. It relates to something else outside of the mechanism of action of the drug. So in theory, it makes sense that UPD would, with ASOs, would potentially respond more robustly, but we don't know.
Jody and Matt, any comments on that issue?
Just one caveat. As you know, the animal model reflects the UB mutations, not deletions, not UPD, and so we just have to be cognizant that all the data that's based on animal studies is not using the deletion and not using those, and so we have to have that qualifier. We're going to be potentially, if the animal models are predictive at all, we can be more confident about UB3 mutations as opposed to deletions. So there's a little bit of a qualifier there.
So moving on to the next set of questions. The next one is, what is expected of any volunteer subjects? And so we started talking a little bit about the big picture of the trial design, but we really haven't talked about the details. So how many visits would be involved? What would be the time frames? Would anybody like to comment on that? I know we're early,
yeah.
but
yeah, we're early in that. But there's kind of typical ways to view this. That there'll be a commitment that will have to be made for people that participate in the trials to come to the centers on the regular basis. Typically, we'll see the a pre-test, an enrollment testing session, a delivery of a therapeutic, and then like a 3-month, a 6-month, and a 1-year, and probably 2-year follow-up. And there'll be a number of different outcome measures that will be measured throughout all of our trials in the centers that are enrolled in the trial. So there will be a large commitment, or there will have to be a large commitment on the people that enroll in the trials. So, odds are you're going to want to we try to recruit people that live within a certain geographic region. We typically try to spread trials out in various locations, so there are not going to be 3 centers in Boston that are going to run the trial. But in general, in clinical trials, that you need to really understand that it's a full commitment, and it will go on at least in our case, 1 or 2 years. And in many of the gene therapy trials, you actually have long-term follow-up where you do follow the patients 10 to 15 years. So once a year, they're asked to continue to follow them.
I think that covers that one. So how will study participants be selected? Anybody want to take a stab at that?
Well, in general, it's all based on the protocol, specific protocol, where eligibility criteria, basically description of who's going to be studied, are very thoroughly described, both in terms of who can be included and who must be excluded. So it's kind of the two sides of the story. Generally speaking, in phase 1 studies, those are fairly restrictive because you really need to assess safety in the most kind of ideal or the cleanest population in terms of you would not want people on a lot of meds. You would not people with a lot of other conditions, particularly this relates to trials in elderly, for example, where there are a lot of chronic illnesses that coexist. So generally speaking, phase 1 studies are looking for a specific subset of patients who, and that subset is defined in the study protocol in a very specific detail. So once the protocol is available and the investigators are selected, and that's, as Jody described, this is really a discussion about feasibility of the study, how long does the study need to be, how large, and that defines the number of clinical sites you recruit. The investigators at each site know the protocol inside out, and they are the best ones to address questions about, well, what about my child, or my son or daughter? Do you think they qualify or not? This really needs to be a discussion with an investigator.
And related to that, where will the clinical studies be done? Where? Any undecided, I guess, at this point? Matt, you're the closest.
Jody already mentioned that a little bit, that the ideal goal is to have them scattered geographically where the largest group, where they're accessible, but also centers that have a special interest, that actually understand clinical trial design. And so these are things that we're looking to have potentially 4, maybe even 5 centers within the US that are scattered geographically, both East Coast, West Coast, and Central. So I think that's really the goal, is really one done both geographically as well as clinicians who have an interest in the space. I unfortunately have to leave.
All right.
I have a plane to catch. And I apologize to you. I would have loved to speak more. We're accessible, and through both FAST and ASF, we will have sort of Q&As for our trial as we get closer. And so those of you who do have specific interest in our trial, we will engage with the community, and we'll put out more specific information and encourage any of you to actually put out questions through the foundations. That's really the best approach to us, and we're more than happy to engage, in fact want to engage and encourage your input as we go through this process. So thank you again for the invitation and being here.
All right, thank you very much, Matt. Sorry. The next question was, how will the therapeutics be administered? So, Matt has to leave, but he already mentioned that the gaboxidol is an orally active drug, so that will be just a simple, pill formulation, relatively traditional drug. Eugene and Jody, would you like to comment on how the different approaches are administered in your cases?
Yeah, I can only speak again on the basis of a single program, which is SMA, for a rare neurologic disorder, and for an NASO in that disorder we are administering intrathecally, which is essentially very similar to an epidural procedure. It is done in a hospital. It's not it is outpatient, but it has to be done by a trained neurologist or anesthesiologist in a hospital setting, and generally lasts between 15 and 30 minutes.
Jody?
Yeah, that still needs to be worked out, but it's very likely it'll be a similar type injection, intrathecal, which is similar, like you said, to an epidural. There are a number of studies that are going on right now, and the very first, one of our advisors was part of the very first study of a gene therapy with an AAV direct injection to the brain, where they did direct injections to the brain, and they're going on now in Batten's disease and other diseases. So it's actually very common, relatively speaking, common to do CNS type delivery that way, and the intrathecal way is essentially goes into the spinal column, and then your cerebral spinal fluid takes the therapeutic up into the brain. So it's a somewhat of an easier way to do it so that you can get into the same location. And as we work on developing our vehicle or our virus, we'll work on getting the virus so that no matter where we ultimately inject, that the virus goes to those cells or those neurons that we need to treat. So those are the things that you work out and ultimately and negotiate when you're developing your trials. But the goal is always as least invasive as possible.
So the next two questions are related. If these treatments are demonstrated to be safe and effective, will they need to be taken, monitored for a lifetime? And on the other side, if you stop taking them, will they stop helping? Will they no longer work after they've been stopped?
Another $100,000 question. We just don't know. We don't have enough experience with this sort of therapies. We believe that it makes sense that these therapies would need to be administered for a while. Whether it's lifetime or not, we don't know. And it may be different for different populations. If you start with a very young child who still has a lot of plasticity, it's quite likely that, or it's possible, I should say, that you may not need the treatment on board once the child is an adult. But we don't have the information yet to speak definitively on that. Even in SMA, which is, again, there's a lot more experience, we're still definitely continuing the trial. The intention is to continue giving therapy to patients who continue to improve.
We also don't know. Gene therapy is typically thought of as one administration, and then it infects the cells and becomes active. We believe that it's likely that, depending on how young, the younger you are, that the more likely over time that it might reduce its effectiveness. And to that end, we look at ways to minimize any effect of redosing or having a second. You're not going to get a gene therapy on a monthly basis or a yearly basis because you develop antibodies to it. So over time, there's been a number of investigations on, do your antibodies potentially go down over time? So if you needed a second dosing, that you could potentially receive that. Additionally, you could potentially change the vehicle or what you encapsulate or how the delivery mechanism, so that you could potentially get. Ultimately, the idea is you give a gene therapy, it goes in the cell and becomes active and stays active. But we have to think. There's a company that's worked in the eye, and it's been 3 years, and it's fading somewhat. There's been a dog that had hemophilia that has been, I think, 12 years with just one treatment. So there's evidence in some of the literature and the models that the therapy lasts a long time, but we just don't know. The field doesn't know how long a long time is, but everybody is very aware of what would be the issues to redose and what can we do in the first dosing to make it so that you have the potential to get another one if you need it.
Are there anticipated to be any side effects?
Well, I can tell you, if I've been doing drug development for a long time, I haven't seen a single drug without side effects. What I can say is that what we care about is the degree and the ability to minimize side effects and learn as much about what is the safety profile in our early stages, particularly before we move into human trials. And that's certainly what FDA is very keenly looking out for. So they need to know what the profile is in animals, and if there is some safety signal, how easy is it to detect? How reversible is that signal? Can you just stop the medicine? Or, if it's a headache, can you treat it and it goes away? Versus something that lasts for days or months. So there's I guess the short answer is there are always side effects. What matters is how predictable they are, how much you learn about them, and how much can you anticipate and reverse them. I can tell you again, with the only experience I could speak of is SMA. The side effect profile that we are seeing now is extremely favorable. We are seeing reactions in some patients who develop headache after an injection or a back pain. Again, it's been relatively easy to treat by pain medicines, and we haven't had patients who had to stop because the side effects were debilitating or worse than the benefits that they were seeing. So again, it's something that is always monitored throughout the development program, and even after the drug is approved, it continues to be monitored by everyone, including the FDA.
Let me go on to the next question. So, and I love that I think we all like the way this one was phrased. What is an honest timeline for clinical trials? So I know both of you have, in your earlier presentations, not made any commitments to start dates for your trials, and I think everybody, at least from your feedback, I think we all recognize that we have to get that IND filed, and that will be a critical event. So maybe you could comment on, once the IND is in place and approved, what is a typical timeline from there for phase 1, and then transition to phase 2, and then transition to phase 3, and then eventual approval of a drug? Maybe you could comment on that aspect
Sure.
of the timeline.
Sure. Once the IND is filed and approved, our plan, our vision is it starts the day after. In all of our developmental timelines, and certainly when you work in industry, the developmental timelines is you're doing all the pre-work you need to get in place for starting the trial. So once the IND is approved, we essentially need to start up the next day. Now, that could mean that we have our sites selected, they understand it, now we can turn on the recruitment faucet, so then we start recruiting patients, so you won't necessarily dose somebody the next day, but it's very quick after you get the IND approval, because the company is working through getting all those things set up and in place before that's approved. And then it's a matter of enrollment time. My instinct is, with this group and with rare diseases in general, that the pump has been primed, because we come and talk to you early, and all of your leadership in the different organizations know what's going on, and you've known about it and you've learned about it. So a lot of you will be interested before we actually get to the IND. So I don't think recruitment timelines are going to be very long to get subjects. In many of the studies, you, particularly dose escalation, which I think you've heard of, you start with a certain dose and then you up the dose and you up the dose. And in those kinds of studies, you typically dose one person and you wait a period of time. So it'll probably be one dosing and you wait a month, and then you can dose the rest. It's sort of the last kind of safety check in that. So it's probably a 1-year phase 1 study for the primary endpoint, which is safety, and following on to year 2. And then depending on where the outcome measures are, you can pick up the phase 2-3 or the pivotal study around the second year, probably after the start. So you end your phase 1 in a year, you have your safety, you go back to FDA, you've analyzed it, it's been evaluated, and then you show that was fine, and in the meantime, you're working on the protocol for 2-3. So it's once you start, it's a several year process, but it feels like probably you're making more progress because you can actually see it. You know people that have gone through it. But it's several years from getting approval of an IND to getting completed through a marketable drug is typical.
So the next question is, are you able to collaborate on outcome measures? And I know there was a lot of discussion during the scientific talk about this.
Yeah, I would say it's a must in rare disease. Again, I spent a fair number of years. You have to collaborate on outcomes measures, despite the fact that our drugs have different mechanisms and potentially address different parts of the disease, but it is, in rare disease, it's absolutely necessary to work with the community of researchers and patients all along the way in refining the outcomes, making sure that the outcomes are sensitive and appropriate for certain groups of patients, because as Matt mentioned, what might be appropriate for a 2-year-old is certainly not necessarily appropriate for an 18-year-old. And it's not just a matter of age. There are different outcome measures have different amount of precision, ability to detect change in a reliable way. So I think we absolutely must be approaching this as what does the community need in order to make any therapy in this space succeed and be able to detect an effect if the effect is there. So it's been done very successfully in many other diseases, including SMA, and I can speak to, again, to that success being really, to me, a very good example of collaborative spirit in rare disease communities such as this one.
Yeah, I'd like to comment on that as well. I mean, I think this is the part that I find very exciting that we can work on today together. While the scientists continue to work on the animal studies and refining the genes, I think the community can continue to work on the outcome measures. And so that's everything. And for the symptoms that you will have problems with. So for seizures, for sleep, for all the different symptoms of Angelman, for the cognitive, the learning, the memory, ideally there would be a very a validated measure that every patient will go through, and that at every time frame. So maybe you go see your neurologist on an annual basis, and you have the same battery of tests every year, and they become validated. So whether it's the three of us or the next pharma company that comes along, there's already a group of outcome measurements there in place, so that now we can start comparing to see if there is an improvement, or how do we measure an improvement. And I think Matt brought up a good point that because there are no current treatments for Angelman syndrome, no one yet knows or has gone to the regulators and says, we believe this is an outcome measure that will show enough efficacy that will get approval. So the more data we have going in with different outcomes on different aspects of the disease, the more likely we'll be able to show the statistical significance to get the meaningful outcome approved. And so I think, yes, I think collaboration, and I think as pharma companies as well, and in our experience in the Friedreich's ataxia field, we are on a collaboration with all the other companies working in this as well, helping to gather that information.
Thank you.
Can I just add just one more remark? And I think it's really exemplified in Angelman's. Regulators recently, or last few years, they're particularly interested in the impact of treatment on the caregivers, not just measuring how many more steps a patient can make, or whether their strength improves, or whether cognitive ability is better, but really what that translates into in terms of impact on caregivers. And in particular, this is true outside the U.S., where oftentimes the payers and regulators outside, in Europe and Asia Pacific, are keenly aware that the drugs have to do more than just allow measurement of some very specific changes on some very artificial scales, be that a change in EEG, which very well may mean something to neurologists, but if that doesn't translate into something that either patient feels or the caregiver detects, they're not so keen in being as excited about these changes as some of the drug developers are. So this is just, again, a plea to think about, as they're called, patient-reported outcomes or caregiver-reported outcomes, as a necessary area of research in development of outcomes, particularly in a disease that is so diverse as Angelman syndrome. So what may change in one patient may not necessarily be the symptom that changes in another, but nonetheless, the impact may be just as significant to the caregivers.
So the next question deals with, what can the Angelman syndrome community do to help each approach progress as quickly as possible to clinical trials? When you get that IND filed, or when you're ready to file it, what should we do? Should we be camping out on the steps of the FDA should we be calling our congressmen or congresswomen to move these things along?
I would urge you not to do that, please. No, I think look, we said that there is still time and probably significant time until the point of IND approval, but this time needs to be used productively. And the last two questions about meaningful outcomes and what was the previous one?
Timelines for trials and side effects.
No, there was
Anyway. Okay,
right,
yeah.
the value of collaboration. I think those two issues are critically important. So being able to build a cohesive community of patients and patient advocacy groups that are very supportive of one another and also understand real issues that the community is struggling with, as well as the research community that is united and understands what the real issues are and can articulate those issues to us, because our ability, we are removed from patient care. It's been a long time since I've treated a patient, certainly a patient with Angelman, so we need to hear from clinicians on what things need to be measured, how frequently, what are the things that are important, and what are the things that are not necessarily as important. So that kind of conversation is invaluable. And as we are getting closer to the IND date, hopefully our knowledge about what that human trial and human program will look like will increase rather than we don't want to find ourselves in a position to 2 years from now, for example, sit and have the same conversation about, well, what are the best outcomes when we are potentially, months away from an IND. So really, let's use this time now productively to work together to outline, first of all, identify what the real gaps are. There's a lot of research that's been done already, and that's great. But what are the real gaps in terms of the need for the future clinical programs? And once those gaps are identified, let's try to fill them, and let's try to work together to do that.
So the next question is dealing with natural history study data. I think you're both familiar with the natural history study that was active for a number of years. Is the collection of additional longitudinal data worthwhile? Are there any other surveys that would be useful to do as we prepare for these upcoming clinical trials?
The short answer is yes. This goes along with the outcome measure that we were talking about. Whether you call it natural history or outcome measures or just following people over time, I think the more controlled data that can be collected pre any clinical trials, the better. You need to understand, like our gene therapy target, Dr. Weaver showed learning and memory in his mice. What are the standardized ways to measure learning and memory in an Angelman syndrome patient? Well, that's probably very different from the 3-year-old to the 10-year-old. So in that regard, a natural history is essentially measuring learning and memory every year or every 2 years or something along that. There's also a lot of evidence on, particularly in a gene therapy, there's unlikely going to be a placebo-controlled study group. So there needs to be some kind of study group to compare the treatment arm with. So if there's a lot of natural history data, a lot of outcome measure data collected on various aspects of the disorder, then that is a much better comparator and it gives us a better idea on how to move forward. So I would say whether it's natural history or outcome measure evaluation or whatever you call it, it's what we've been talking about in that getting different measures at different age populations so that you're prepared for our trials and for any other trials that come along. Clearly, as someone had asked me before, well, we might get those wrong. Well, sure, you'll pick a series of measurements now, and in 5 years from now, you'll say, oh, I need to add this one in, or I'm going to drop this one off. But it certainly gives you a good place to start to have a data set that you can look across the lifespan, the age span.
The last of the submitted questions, I think this is a very intriguing one. Parents and caregivers are very busy, very active. They don't necessarily have the scientific expertise to evaluate all of the different reports and trials and things that are being discussed. So the question here is, what advice would you give to parents and caregivers in assessing the value and credibility of the research findings that are out there?
So I think it probably goes with any kind of medical issue that you might have for yourself or your children or a family member. You'll hear of something. Typically, what everybody does is they start with Google. So you pick up as much knowledge as you can. Then you talk to people that you trust and that you respect and that you can believe in. And so you listen to, maybe there's a new therapeutic out. So you listen to the company talk about the therapeutic. You see if they talk to you in the way that you can understand. Then you talk to your immediate family or your friends. You talk to your primary physician that you respect and believe in. You go talk to your neurologist, or you talk to friends of your family. So I think you can't possibly, none of us can possibly keep up on everything that's going on in medicine for all the different aspects of all our lives. But you have a group that you trust and respect and believe in that you can discuss those ideas with. And I would say lean on your main care provider that is very tuned in. The scientists that were at the scientific meeting and the clinicians are very tuned in to what's going on and have really analyzed. So go talk to them about what might be coming out. And then staying connected to your advocacy groups. They are very up on everything. And in fact, many of us have long conversations with the advocacy groups before we ever announce anything about what's going to happen. So they know what's going on in the background. So you can get as much information from them, and then it's not biased by a Google approach. It's presented to you through people that care about the same disorder you do. That's what I would do.
Yeah, I completely agree. I would also emphasize getting to know your foundations and don't be afraid to ask them. They have scientific boards who are able to interpret complex studies or complex scientific data, and they are able to provide the digest that is more friendly to understand to the non-scientists. That's their jobs oftentimes, and I've seen foundations do it very, very successfully for many other rare diseases where there's not a lot of experts, including clinicians. I mean, a lot of parents are much better experts than their doctors when it comes to rare diseases. So foundations are key to be able to support you and in understanding where the science is going in this disease.
Okay. So that's the end of the submitted questions. So we have a microphone in the aisle there. I'm sensing that everybody is exhausted and ready to go to the reception. But if anybody has any questions, please just move to the aisle and use the microphone so that everybody in the room, as well as everybody on the internet, can hear you.
Thank you. Thank you for your presentation and for your interest in Angelman syndrome. My question is, what endpoints are you expecting from the two research, Gaboxadol and gene therapy? And also, are you incorporating antidotes for Gaboxadol and gene therapy? And are they going to be included in the research? in case side effects happen and if they're severe?
First of all, let me. Unfortunately, Matt had to leave. I'll try, from my background, to comment on the gaboxidol side. With respect to side effects and adverse events, gaboxidol has a relatively short half-life. He talked about it today that in the blood and plasma, it only has about a 90-minute half-life. So if there are any adverse effects, it's going to wash out on its own very, very quickly. It does get, once it gets into the brain, it does stay in the brain a bit longer, but it is still it's hours to wash out or to be metabolized and get out of the body. It's a very small molecule, and so metabolism is very quickly. Having said that, the issues with the other two.
And I thought that was a fantastic idea. So I'm going to take that back with me, and we are hoping to, we say they're press releases, but not necessarily press releases. We can just do updates, website releases that tell you this study was completed and, it showed this or something, like that. But absolutely, yes. And like I said, we want to be very collaborative, so you can always ask us. We plan to be at all the events as much as possible, so feel free to reach out to us along the way. And we'll get busy and probably not communicate enough, so we are happy for you to ask us questions.
I completely agree. I would also add that at ISIS there is patient advocacy group. Basically, there is a team of two. I mean, we are still a small company, but Christina, who's in charge of that group, I think she knows every family with SMA in the world. It feels that she is so engaged and certainly connected with advocacy groups, but even individual families who need to know more, who wants to be engaged and get updates. This is also a very good mechanism to stay in touch if you're interested in our progress. And she's phenomenal.
My question is, I'm thinking about 5 years in the future when something's been developed specifically for the American market. Do you have any thoughts about the cost to the consumers of your therapies? Because even as of right now, a lot of families are struggling to even afford seizure medications and spend lots of time fighting. They're stuck between their pharmacies, their neurologists, and their insurance companies. So I was just wondering, I realize that with your companies are putting lots of money into the research. You have to recoup that money, and again, this being a rare syndrome, how is that going to, on the end of the product, what does that mean for American consumers?
That's an excellent question, and certainly very relevant to rare disease programs, because let's be honest, a lot of rare disease, when they come to market, bear pretty significant price tags. What is usually done is that in anticipation of approval, there's a lot of work that goes with the payers to explain the value of the drug. In almost all of these rare diseases, these medicines are really transformative. They are not just kind of minor improvements, they are meaningful improvements. And that gives us leverage to work with payers ahead of time to say, here's the program, it's a rare disease, the drug really needs to provide enough revenue to offset the cost. But because it's a rare disease, it's not a huge ticket item to you, Blue Cross Blue Shield. I'm just kind of picking on a pair. And it, by the way, is not just providing a very minor increment over what's already available. It's something that either is first treatment for the condition that hasn't been treated before ever, or something that really provides very meaningful effect. And that gets back to the outcomes and the importance of really developing something that captures the real value of these drugs to families, to patients, not just measuring something that is a little bit more esoteric for the payers. They don't know what improvements in EEG signal means, right? So Long answer is yes, the costs are probably going to be higher than seizure meds, but it doesn't necessarily mean that it's going to be, affecting the copay. It does usually involve a lot of negotiation with the payers because these are rare diseases and they're much more willing to eat that cost because overall it doesn't affect their bottom line as much as a diabetes drug or cardiovascular drug.
No, we're definitely not going to hear anything. Yeah, I agree. That's fine. Real loud. Yeah.
I'll yell over there.
We can huddle.
Yeah.
So, I was going to say, how about I start? So the question I have revolves more around acquiring structured and unstructured data in a developmentally disabled population, the challenges that you see and what challenges or what tools could be developed, what gaps exist, where tools could be developed to overcome the problems.
Yeah, yes, it's difficult to understand to capture this data in. Oh wow. Hello. Yes. So yeah, it's difficult to.
Maybe you can repeat the question.
Oh, okay. The gist of the question was that in the developmentally delayed population, it's very difficult to capture structured outcomes or structured measure over time. And yes, absolutely, 100% agree with that, as I've heard the last few days. And so that's where we need to look to our educational colleagues, our scientific colleagues, and that's where the cross-disciplinary communication and teams really matter, because the educational specialists, the speech pathologists, the occupational therapists, the physical therapists can work with the scientists and understanding to develop those measures, and that I think it needs to happen, and I think it's happened in other diseases. So that I think that's where the collaboration among you all with the scientists can start making that happen.
And this is where it's really important that these measures are not developed by the drug companies. It's really something that is practical and thought through and developed by people who are really experienced in treating these children and assessing children or adults on a day-to-day basis. It's certainly been done. It remains a challenge in SMA and other pediatric conditions. It's not just developmental issues. In children, young children, it's an issue of endurance oftentimes, whether they're able to get certain measurements at 2 o'clock in the afternoon versus 7 a.m. So all of those things that seem simple and intuitive are important, and they need to be thought through very, very carefully and discussed with people who take care of these patients day to day.
All right, I think this may be our final question, so please go ahead.
We greatly appreciate as parents and caregivers all of the information that we've learned today. One, hopefully, maybe not a last question, but at least you identified that there are other studies that are taking place now or in the very near future, in addition to the ones that have been presented this evening.
So if I understand the question, was are there other programs that will be advancing in the clinic in the not too distant future?
Specifically referring to AS, yes.
Yes. Would either of you like to take that, or would you like me to go after that?
I think you should. I mean, I don't know of it. I don't know of any, but you might know more than I do.
Right. So there are, in a broad sense, there are a tremendous amount of programs that have not reached the state where we have identified targets, downstream targets from the UBE3A gene that are potential targets for therapies. There are compounds that are known to interact with those targets, and there is interest in exploring those, and we need more academic investigators to push those forwards, and we also need more pharma companies to then get excited about exploring those and pushing those on into the clinic. So I think what we're going to be, and going back a couple of years, 2 years ago at this conference, at the last conference in 2013, I indicated that what we should anticipate is that in the next 3 to 5 years, we will see a whole series of compounds and programs being ready to advance in the clinic. I think we've talked about 3 today. Those are the ones that are publicly, these companies have, formally made a public announcement about their activities. There are a lot of other companies that are exploring things, and there are a lot of other academic investigators that are moving programs forward. So I think we will see in the next 3 to 5 years a whole series of programs that will be of interest in exploring clinically. Some of them will pan out, some of them will not, but there's going to be a whole lot more coming down. We have a pipeline of programs, but a lot of them are at an early stage and are not ready to go into the clinic yet, but will be in the next few years.
Very good. Thank you.
I want to say, being from California, I think in the time of this meeting, we have gotten more rain here in Chicago than the annual rainfall in San Diego. Yeah, we've doubled it at least. So I just want to thank all of you for your patience. And thank you for your participation. I think overall it's a very, very positive time for research, and there's a lot of hope for success in the future. So I want to thank you for your attendance, and let's thank our collaborators again. Thank you very much.